And what we're seeing is increased cofilin complex formation in the disease process for 3 out of those 4 patients. The only one that didn't, so in this case we're going from, this first patient, he went from stage 0 to what they call stage 2. You can see a huge increase. You see increases in these 2 patients as they go from pre-symptomatic to stage 1. And we didn't see a difference with 1 patient, but apparently there's some controversy whether or not that person was diagnosed properly at which stage. So that needs to be revisited. And I think this is giving us tremendous promise for a biomarker in the clinic, anyway. And now to feed back from the bedside back to the lab, we now think if we're going to really identify this as a potential biomarker, it's got to be able to show similar functions and similar readouts in animal models. So we're looking together with Jill Bates in longitudinal samples from the R62 mouse and from other animal models, including Anthony Chan's rhesus. So this could mean also that there are potentially new targets for cofilin-mediated neurodegeneration and Huntington's disease. The reason why cofilin rods form due to stress is due to low ATP levels in the cell. When ATP levels are decreased, slingshot phosphatase is activated, which activates cofilin, and this causes arrest of all actin polymerization, which releases up a tremendous amount of ATP. It's estimated that about 75% of the total cellular ATP is being used by actin remodulation in a neuron. And the off switch to this is limb kinase, which activates a very interesting protein called profilin, which has been very well described in the Huntington community as a direct Huntington binding protein. So when you look at the immunoterminus of Huntington, Huntington contains direct and indirect actin binding motifs. The polyproline regions are known to bind to profilin molecules, and if you look at crystal structures of polyproline with profilin, one stretch of polyproline can bind 2 molecules of profilin, so there's probably a 4-to-1 ratio in the Huntington protein. In the middle of those pure polyproline stretches, there's a proline-rich domain that Plomin's group has described very nicely, and we've reproduced in our lab as interacting domain for an interesting protein called paxin-1, which just happens to be a tetramer. And both paxin-1 and the immunoterminal domain are substrates for PKC alpha and CPT. And right beside the polyproline region, depending on which search algorithm you use, there are 8 consensus spectrum and actin binding motifs in Huntington. So our proposed model that we're working on so far is that there is a direct role in actin remodulation in the nucleus. In response to stress environment, cofilin gets activated, caps actin, frees up a tremendous amount of ATP. The off switch is Huntington being signaled from the ER in this process to localize Huntington and profilin to these cofilin rods. And profilin is the opposite effect of cofilin. What profilin does is increase actin branching and growth. And the net result that we see is the release of cofilin from the rods and the very quick disappearance of cofilin. But in the mutant situation, Huntington tacks onto these rods and stays there. It's stuck there because of the polyglutine expansion. What we're seeing is increased cofilin, increased dimers. We're seeing persistent rods, and then we suspect this is leading to homeostasis of energy effects and transcription effects. So, what is the role of actin in the nucleus? This is very controversial. But in order to put this into a context, it's a very preliminary data. There may be a role of histone deacetylases in the modulation of nuclear actin. This is an image of human GCN5, histone acetyltransferase, overexpressed in a human cell. And this is what happens. This is actin. And if we make a point mutation in GCN5 that destroys its histone acetyltransferase activity, we don't see these filaments. And that says that histone acetyltransferases may also be able to modulate actin acetylation and polymerization in the nucleus. So, in the future, what we really want to ask as questions are like, what does the role of antioxidants have in the neuroprotection of Huntington's disease? And I don't think this has been looked at properly in the context of HD. What are the roles of potentially the CK2 inhibitors in HD neuroprotection? And in particular, we're going to be starting mouse models to ask exactly what does the role of nuclear localization have on neurotoxicity now that we have a way to control out of the nucleus. And can this also be done by small molecules? And I think a really important question from a lot of this, and I've been talking a lot to James Bamberg about this, is there a potential common mechanism between HD and what I think is going to be a subtype of Alzheimer's disease patients involving oxidative stress? So I've just been told to plug this in the last minute. Welcome to Vancouver in 2009. Mark this date in your calendar, September 12th to 15th. Michael is very open to thoughts and suggestions about how to run this Congress. And he invites any of you to get in touch with him with any ideas. And he's really looking at doing things differently for this. And I'm one of the organizers as well, so I'll look forward to seeing you there. So here's a lab, outstanding in the field. And particularly the work on the NLS was done by Carly Desmond. Lise Muncie is here, so you can talk to her about the fulfilling rod story. And Randy is also here, so you can talk to him about 18 and 18 rod. Thank you. So, questions?
Well, Rick, Xiaojiang from Emory. So the identification of that PY and LS domain in Huntington is very interesting. But several groups, including gel-based, Wikimedia, and all groups, to use the fractionation can only detect a very minimal forest Huntington in the nuclear fraction. And also ASP2 mice which express exon 1 and N131 transgenital animals which express transgenital protein that do not have this NLS domain can still accumulate in the nuclear eye. So how can you comment?
Okay. So the first question is about the fractionation. And it turns out the step in everyone's fractionation protocol, and which is something that Randy fought for 6 months, and the results of his paper, is that the minute you put cells on ice, you are redistributing Huntington in the cell. You cannot put cells on ice. So these fractionation protocols take place at room temperature on a very long day. So putting your cells on ice makes a huge difference on what you're going to see by biochemical fractionation. The second question is, all right, so if these small fragment models of exon 1 and 171, which show enhanced pathology, but yet don't have a nuclear localization signal, and we see nuclear protein, how is that happening? The reason is they're just very small. And in fact, what I think the reason may be for the enhanced pathology in those, and one reason for the enhanced pathology in those fragments, is that in a larger fragment, we would have control, normal control of Huntington nuclear entry. But if you remove the NLS and you make a very small fragment that can just diffuse into the nucleus, because those, all of those fragments are far below the diffusion limit for nuclear entry. We're just getting uncontrolled nuclear entry, and we're getting just uncontrolled ability for it to accumulate.
So I have just a short comment. I just wanted to say I don't really think there's any fundamental difference between what the two of us are saying about the N-terminus confirmation. The way I would phrase it is that the N-terminal sequence has a propensity